rolling here, so please take a seat. Since we've had a big turnover here, I'm going to reintroduce our next speaker, William Benedict. My name is Fred Henson, and I'm your moderator. Folks sitting in the back row, if you're having trouble hearing, just make this signal and I can amp up the microphone. So without further ado, William.
Yeah, I'll try to talk into the mic here. Hey, my name is Billy Benedict, and I'm going to be talking about some work that we've been doing for the past couple of years in Hudson River Tributaries to hopefully inform some future restoration projects. So a little background on the Hudson River watershed, if you're not familiar with it. Main stem and tributaries combine to provide thousands of river miles of aquatic habitat to the East Coast. But that habitat's largely fragmented by both natural and geomorphic features and anthropogenic barriers. The first barrier on about half of those tributaries is a waterfall, and the remaining tributaries, the first barrier is a dam. And dam removal represents one of the best options to reconnect habitat. So this project really spawned from conversations that were happening at Riverkeeper when they kept on losing out on federal funding for their dam removals. Because Hudson River tributaries are so serially dammed, you can remove a dam, but you might only get a quarter mile upstream before you reach the next dam. And when you compare that to some systems on the West Coast where you remove a dam and get 50 miles of upstream habitat, it's really a no-brainer who's going to win out on that funding battle. So we kind of brainstormed to quantify ways to try and see what the ecological return would be for a certain dam removal or restoration project. And bringing that to the table would hopefully make the Hudson River more competitive when it comes to getting some federal grants for restoration projects. So the goal of the project was to characterize the ecological value of Hudson River tributaries through metrics of biological productivity across different trophic levels as well as across a range of human impacts. We did that by sampling upstream and downstream of the first barrier in 10 Hudson River tributaries, all south of the federal dam in Troy, that being the Poston Kill, Winans Kill, Hanacroy Creek, Saw Kill, Black Creek, Fall Kill, Wasaya Creek, Furnace Brook, Cedar Pond Brook, and Minneskiango Creek. So starting at the bottom there, we got estimates of ecosystem metabolism, then maybe jumping up a couple of trophic levels, we sampled the macroinvertebrate communities, and then we modeled potential blueback herring production potential in the tributaries. So quantifying productivity at the base of the food web was done through estimates of gross primary productivity and ecosystem respiration, which combine to be your stream metabolic rate, or the rate at which organic or atmospheric carbon is fixed by plants and algae via photosynthesis. And on the flip side of that, the rate at which that organic carbon is consumed by everything in the ecosystem. You can do that a couple different ways, but ultimately you're taking measurements of dissolved oxygen in the water. So originally we tried the dark light bottle method, which a little pilot study that we did was a terrible fail. So we went hunting for alternatives and found a cool R package called Stream Metabolizer, which inversely models values of GPP, ER, and the gas exchange rate that best fit your modeled and observed DO curves. So we went out, bought some hobo loggers, deployed them at upstream and downstream sites for up to 4 days, and then fit Bayesian state space models that through the Stream Metabolizer package that take into consideration both observation and process errors. So these are some plots that you get from the Stream Metabolizer package. This is from the upstream site at Minneskiango Creek. On the left there you have 3 days of GPP and ER estimates with 95% credible intervals. And on the right there you have your modeled and observed DO curves. The lines being the model DO and the points being the observed DO. And going top to bottom there's the DO in milligrams per liter, the DO percent saturation, and the rate of change in DO. So GPP estimates for all 10 of our tributaries. The panels there are each individual tributary. The y-axis is GPP. The x-axis is downstream and upstream sites, with downstream being those light gray points, black dots being the upstream sites with 95% credible intervals for everything. But generally, the estimates varied among tributaries and between upstream and downstream sites. However, we did see more productivity upstream of barriers, and also tributaries west of the Hudson River were more productive than tributaries on the east side of the Hudson River. So that would be Black Creek, Cedar Pond Brook, Hannaquai Creek, Minisciongo Creek, and Quisea Creek. So then to quantify a lower trophic level consumer, we sampled macroinvertebrate community. They're commonly used as biological indicators because they're easy to catch. They live most of their life in water, and they have varying sensitivities to both water pollution and habitat disturbances. You can also sort them into functional feeding groups, which can tell you a lot about basal productivity and your ecosystem function. So we used a Cerber sampler, which is pictured there. We sampled in a riffle, run, and pool site at all of our sites, so that's 60 samples. Everything was identified and enumerated to get abundance and diversity. Then we also dried them and burned them in a drying oven and muffle furnace to get dry weight and ash-free dry weight for biomass. So this is a plot for numerical abundance with count on the y-axis, tributaries on the x-axis. Each panel is habitat, with downstream being the gray bars, upstream sites being the black bars. The general trend here is that abundance was higher in riffles and lowest in the pools, with no clear trend in upstream and downstream abundance. Looking at those biomass metrics of dry weight and ash-free dry weight, similar trend with riffles having the most biomass and pools having the least amount of biomass. But here we see, in general, upstream sites having higher biomass than downstream sites. And then going on to diversity, we looked at family level richness. Same trend here, but a little bit less variable, with riffles being most diverse, pools being least diverse, and no clear trend in upstream and downstream sites. Finally, a colorful plot for you guys, no more black and white. This is the relative abundance of functional feeding groups at all our sites. We're still kind of trying to sink our teeth into this and make heads or tails of it, but you have the CF and CG are your collector filters and collector gatherers. Those are like your net spinning caddisflies or your mayflies. They're eating fine particulate matter. PR is your predators like Megaloptera that are eating other aquatic insects. SC are scrapers like water pennies or snails that are scraping periphyton off of wood and rocks. And then SH is shredders like stoneflies that are eating more coarse particulate matter like leaf litter. And other is just macroinvertebrates that couldn't be identified to a low enough level to sort them into a functional feeding group. And yeah, like I said, we're still trying to digest all this information, but in general it seems like the collectors and the scrapers are the dominant feeding groups in our sites. And then finally, to estimate productivity of a higher trophic level consumer, we looked at blueback herring. So alewife and bluebacks are collectively known as river herring. They're anadromous, so they come into fresh water to spawn. Every spring, hundreds of thousands, if not millions, of river herring come in, bringing with them ocean-derived nutrients and productivity into the tributaries. And the latest, the 2024 benchmark stock assessment had newly developed habitat models that we use to assess habitat availability and the production potential of blueback herring in our tributaries. So yeah, we used those newly compiled habitat data and coupled it with stochastic population models developed by Dan Stitch and colleagues, Nick Jordan, he's here too. And that's in the anadrofish package in R. And we looked at two different scenarios, one being a no passage scenario and one being a no dam scenario. So no passage is basically taking all of that habitat up until the first dam and not including any upstream habitat. And then no dam scenario is kind of like a hypothetical scenario where no dams exist and blueback herring have all the available habitat that's known for them. So here's a plot for the production potential of spawning adults. Spawning adults on the y-axis, tributaries on the x-axis with the no dam scenario in the light gray, no passage scenario in the dark gray. Tributaries were the first barriers of waterfall, didn't get a no dam scenario because it wouldn't change the amount of available habitat for them. Take the Fog Hill, for example. If you have river herring coming into a tributary, even if you remove all of the dams above that waterfall, they're still going to hit that waterfall and not be able to get above that. But for tributaries that did get a no dam scenario, that being Cedar Pond Brook, Furnace Brook, Minneskiango Creek, and Quisea Creek, that newly available habitat results in an increase in production potential for blueback herring. And excluding Furnace Brook, that increase is an order of magnitude greater. So to sum up, we got GPP estimates that varied among tributaries, but in general, upstream sites were more productive, and these estimates were similar to historical work, and that spatial trend of west of Hudson River tributaries being more productive than east of Hudson River tributaries is also reflected in the literature. For benthic macroinvertebrates, all of our metrics of abundance, diversity, and biomass showed the same trend of being highest in the riffles and lowest in the pools. However, higher biomass was seen upstream of barriers. So that's kind of suggesting to us that lower trophic productivity might be blocked upstream of dams, preventing migratory fish from accessing that more productive habitat. That's kind of echoed in our river herring modeling, where we see if you remove those dams and allow those river herring to have that habitat, you'll see an increase in productivity. So we still have more work to do. We're still digging into those functional feeding groups. And we also want to look at some land use data. So kind of seeing if there's any correlation between like canopy cover and our GPP estimates or developed land and our macro metrics. That'll be really interesting to look into. And then we also did paired eDNA and electrofishing surveys at all of our sites, kind of like the work that Scott did. And those data will show a really clear picture of the sort of effect that barriers have on these like top level predators in these systems when you're catching 800 eels downstream of a barrier and then you go right above it and you catch 2 eels. So we're excited to share that when it's ready. Then yeah, I'd just like to thank WRI and the Epley Foundation for funding this, the Biological Field Station at SUNY Oneonta for letting me use their facilities and their equipment, Riverkeeper and the New York DEC for their continued support, especially the Hudson River Fisheries Unit. See a lot of y'all in the room, thank you. And also want to thank all the volunteers that helped out. And with that, yeah, I'll take any questions. If I can't answer them, Dan Stitch is in the back. Yes, he'll answer it. Yeah.
You started by comparing the total length of western waterways versus the eastern ones between the dams. Have similar GPP estimates been made out there, and how does yours compare?
Yeah, so they're similar. They're similar to like nationwide estimates of similar sized tributaries.
The box plot of the no passage and no dam, right?
Yep.
There's 3 river systems that showed that there would be a predicted increase in spawning of one.
That's
So
correct.
just, that's, like if you summed up all the grays and then summed up all the light grays, what proportion would the light grays, like how much of an effect would that have on the overall abundance in the system, proportion?
Yeah, it's a good question. I don't know if we've looked at that yet. I think it increased it by quite a bit, but I don't know the exact percentage.
Well, if you like Black Creek there and or Hanacroy
Yep.
Creek, right? Their abundance is really big.
Yeah, so in those tributaries, you have a lot of habitat before you reach that first waterfall.
Yeah, right.
So I think it's just like an artifact of how much habitat's actually in these systems. Yeah.
Yeah, great talk. So if I understood correctly, you said one of the motivations for doing this in the first place was to make our dam removal applications more competitive on a national scale. I mean, is there any reason to believe that, or are we far enough along with this work yet to actually see, reaping the benefits of that?
I don't know if we're far enough along yet, but I know Scott Cuppett, the Fisheries department, is really psyched on the work that we're doing. He's talking with the dam owner on Cedar Pond Brook, and yeah, I just know people are excited about it, but I think it's too early to tell if anything's going to come.
Sounds great.
You discussed the introduction of ocean nutrients into the streams. Did you notice any effects from to the freshwater species or the macroinvertebrates because of the introduction of the nutrients?
We didn't look into that. I know there's plenty of studies about like the nutrients that salmon bring into tributaries and stuff like that. So yeah, I'm sure river herring coming into the millions into the tributaries are certainly having an effect, but I don't know if anyone's tried to quantify that.
Question for you.
Yeah.
In your no dam scenario, did you assume the proportions of mesohabitats would be as is, or did you have some assumptions about how those proportions might change if barriers were removed?
I think it's just a pure like linear like quantity of habitat. So
Okay.
yeah. Thank you.
Thank you.